Hello guys, welcome to my YouTube channel. So today we are going to discuss cDNA library. Now, what is a DNA library in general? A DNA library is a collection of all the DNA sequences present in a particular organism. Now, what is cDNA library? cDNA library is actually the collection of all the cloned DNA sequences that are complementary to the mRNA that is present in an organism. So, in other words, it is the collection of all the cDNAs that will be obtained from the mRNA present in any organism. So, let us see how we can do that. Let us see the principle. So, the principle behind this is that suppose you have an mRNA. So, by reverse transcription with the help of reverse transcriptase enzyme, from this mRNA, you can synthesize a complementary DNA strand. Okay, to this mRNA. Now this complementary DNA strand synthesized from this mRNA by reverse transcription is called as cDNA. So in this way, you can prepare the cDNA of all the mRNAs present in an organism and get an inference of all the DNA sequences present in that particular organism. So what is the procedure? The first step is to isolate and collect all the mRNA present in any organism. So you have collected all the mRNA present in an organism, then you perform reverse transcription with the help of reverse transcriptase enzyme and you synthesize all the cDNAs. Okay, so you synthesize all the cDNAs. Now after synthesis of all the cDNAs that are complementary to the mRNA, what do? You insert it into bacterial plasmid. So in here you perform recombinant DNA technology. How do you do that? Suppose this is the bacterial plasmid and here are the synthesized cDNAs. So insert the cDNAs within the bacterial plasmids at this position, and the plasmid that will be formed is called as the recombinant DNA. So the recombinant DNA that will be produced after the insertion of this cDNA into the bacterial plasmid, what do? You insert these recombinant DNAs within bacteria and you allow these bacteria to grow for certain generations. Why? Because as the bacteria grows, the recombinant DNA within them also grows. So the amount of genetic material doubles. Okay, so after a certain generations, what do? You isolate all the recombinant DNAs and you perform purification to get only the desired DNA from the bacteria, and then you perform sequencing. So this you can do that by sort of various sequencing techniques that we have, like Maxim Gilbert sequencing or Sanger sequencing. So after sequencing, what do you know? What do you get? You get an idea of the sequences present in that particular organism. So cDNA library is basically performed to know what are the different DNA sequences that are present in that particular organism.